Good morning, my dear student. Hope you are doing well. You have already watched my last videos on recombinant DNA technology, its principle, definition, procedure, steps, and the diagrams. Hope you understood. And in this course, plant physiology and biotechnology, the another important topic in module 5 is methods of gene transfer in plant. We were discussing about genes and making transgenic GMO in plants or even in animals or even the microorganisms. Even in the applications of recombinant biotechnology, you can see the development of novel microbial strain for fermentation technology, where also you can insert the more enzyme synthetic enzyme synthesis responsive genes. So here in only we'll discuss about the plant, how genes can be transferred into the plants. So first of all, it is defined simply as a technique to efficiently and stably introduce foreign genes into the genome of target cell. So what does it mean? Foreign genes is a simple technique of foreign genes introduction into the genome of target cell. Into the genome of target cell means your target species, that species where you want to insert the important gene, foreign gene. Okay. The methods are classified into two groups. First of all is the direct method and second is the indirect method. Direct method and indirect method is also called agrobacterium mediated gene transfer disease, and direct method where you can insert maximum number of DNA molecule directly into the target cell. And indirect means via agrobacteria, means a bacterial host cell where via plasmid or vector you can insert the gene into the target species. That's why this is called indirect. So in the direct gene transfer, on the delivery of large amount of DNA particle, in case of direct gene transfer, already I told, large amount of DNA particle can be inserted into the target genome. And it is mostly used for gene transfer in cereal crops, monocotyledons and dicotyledons crops. So cereal crops, you know, already I have discussed about the plant kingdom, what is monocotyledon, what is dicotyledon, and the direct gene transfer include here are several example: particle bombardment, electroporation, ultrasound. I will discuss later. Okay. And agrobacterium mediated gene transfer are based on the utilizing of agrobacterium, a pathogen of dicotyledon, broad-leafed plant, and transfer the gene into the plant genome. So this is the indirect method of gene transfer. So now we'll talk about the general approach for genetic engineering in plant may be outlined as follows. Genetic engineering means is nothing but you are inserting a gene into a target species. That is nothing but the engineering work. And genetic engineering means you are inserting the gene into a target cell so that their setup will be changed, setup will be recombined or recombinant. That's why it is called genetic engineering. You are doing the engineering part. So introduction of a gene of interest into the host cell is concerned plant species. First of all, this is the thing you need to select the gene of interest. Then integration of this gene into the nuclear or organelle genome into the plant cell. Integration means collection of some of the expression of the transferred gene in new genetic background. Then the gene should be transferred into the target species. Then regeneration of whole plants from the genetically modified cells. Then after insertion of the genes, The plant cell should be divide and should develop shoots and root followed by a complete plantlet. Until unless you are getting a complete plant, your experiment is not successful. Transmission of the transferred gene to the sexual progeny of these plants. It may be noted that gene transfer in plants are primarily based on the tissue culture and integration of expression of the produced gene must be stable to be transmitted through the sexual processes. So here thing is that already you have transferred the gene into the target plant species via direct method or indirect method. So after that, what should be done? Because you need to culture the plant via plant tissue culture. Here is the importance of plant tissue culture. Because once you have inserted the gene into the plant, then you have to culture the plant in the artificial condition, supplemented with artificial nutrient medium to the plant species, and then followed by development of new plantlet that may be termed as transgenic. Okay, so this slide is very very important for all of you. Is frequently questions may comes from this slide, means what are the types of gene transfer methods actually in plants. So already I told you. So broadly, it is classified into two categories. First is the direct method. You can see here direct method. Direct method is the physical and chemical method includes the direct method. And biological methods is called agrobacterium species. It is called indirect method. So plant transformation methods is physical, chemical, and biological, right? So now under the physical means you can understand micro injection, like the injection you see in the medical sector. Something can be pushed inside the target sample. Likewise, micro injection you can use in the solution using the micro syringe. You can directly insert the gene into the target species. And pressure or mechanical shock via you can also insert. Ballistic gene gun also, like here the in this gun bullet is nothing but the gene. Okay, so using the gene gun also you can insert the genes into the plant. Particle bombardment means using nanoparticles, silver nanoparticles, gold nanoparticles. Using silver nanoparticles and gold nanoparticles, you can insert the gene via particle bombardment. You can bombard the particle inside the cell via electroporation. Here is another technique. Electroporation is also separately can be experimented. Electroporation means in this name you can understand via electric shock you can lyse the cell wall followed by the cell membrane, and you can insert the gene inside the nucleus. Okay, then silica carbon filters also there, and mediated. Laser means laser beam you can see in spectrophotometer. There are some microwave length laser beam. Okay, laser mediated, although through laser you can also insert the gene into the target plant species. Now in the chemical portion, PEG, using PEG, polyethylene glycol, already we use PEG for protoplast fusion. Protoplast PEG actually dilute the cell membrane. So once the cell membrane is lysed or dissolved, so directly you can insert the gene into the cell. Then calcium phosphate also, they are the chemical compound. Dextran sulfate is also there. Some artificial lipids are also there, and protein dendrimers also there. Protein dendrimers are the star-like biological macromolecules having several branches of amino acids where you can conjugate the genes and can be inserted into the plant species. Okay, so this is all about the physical and chemical direct gene transfer method. Now, in the biological gene transfer method via agrobacterium species, so here the transformation via transformation means the gene has to be cloned in a vector. Already we have discussed in the recombinant DNA technology. And then the vector, cloned vector, means the plasmid, recombinant plasmid should be inserted in the agrobacterium. And then this gene will be cultured in the media. Okay, in the during plant tissue culture, agrobacterium containing media, the plant species will be cultured, then the genes will be inserted into the plant species. Okay, so this is the indirect type of gene transfer methods in plants. So this is all about the highlights of plant transformation methods or gene transfer. Hope you understand, and if you have any doubt, we will clear in the class. Thank you.